Okay, I'm very pleased to introduce our speaker today, Dr. Sreeram Kannan. Sreeram is currently a postdoctoral researcher at the University of California, Berkeley. He received his PhD in electrical engineering and MS in mathematics from the University of Illinois in Urbana-Champaign. He's a co-recipient of the Van Valkenburg Research Award from UIUC, the Qualcomm Roberto Padovani Scholarship for Outstanding Interns, the Qualcomm Cognitive Radio Contest First Prize, the SVC AIA Medal from the Indian Institute of Science, and the Intel India Student Research Contest First Prize. His research interests include applications of information theory and approximation algorithms to wireless networks and computational biology, and we are delighted to have him here. Thank you.
Good morning, everybody. Thanks for coming to my talk. I'm Sreeram, and I'm today going to talk about optimal RNA assembly from information theory to software. So my own training is originally in information theory, and this is a problem where we found a very interesting application of information theory to computational biology. Okay, so this is a customary slide in almost all the bioinformatics talks, and the basic idea here is that there has been a revolution going on. So most of electrical engineering growth has been driven by the Moore's law, the doubling of transistors per unit area and the cost of transistors. There has been a similar but even more profound revolution in computational biology, driven by the reduction in sequencing costs. So here you see that the cost of sequencing a genome has dropped even more dramatically than Moore's law, more than exponential drop. Currently, the cost of sequencing a single genome has fallen to $1,000. So this was in 2011. Now it's $1,000 in 2014. So it's a tremendous revolution. And what this has engendered is not only the ability to cheaply sequence genomes, but also a variety of other biological problems can now be translated into sequencing problems. So this is kind of a revolution in biology, and this has enabled the exploitation of DNA sequencing for doing a variety of other biological measurements in using methods which are called star-seq methods, for example, RNA-seq, ChIP-seq, and so on, which can be used to measure all other different phenomena like RNA, protein-RNA interactions, protein-DNA interactions, DNA three-dimensional structure, and so on. Okay, so I'll start the talk, since this is an electrical engineering audience, I'll start the talk with the basics of what the RNA is. So the way to think of the RNA is it's like the RAM of the cells. So the DNA is like the operating system or the hard disk based on which all the instructions of the cell are written. However, if you want to know what the cell is doing right now, you look at the RNA because the instructions in the DNA are copied to mRNA by transcription, and the RNA transcripts basically capture. So if you look at the RNA in the cell, it'll tell you what the cell is doing right now. For example, why is a skin cell different from an eye cell? It's because it's expressing different aspects of the DNA. So if you look at the RNA in the cell, you'll get to know what aspect of the DNA is being currently expressed. So the importance of RNA sequencing is that once you get to know what the RNA is in the cell, it can be exploited for a variety of purposes. For example, in clinical applications. For example, you want to know why is it that a given cell is diseased as opposed to another cell which is not diseased. You look at the RNA and you will know which aspect of the DNA is not getting expressed correctly. Therefore, it's very useful in clinical applications. It's also useful in the discovery of novel functions. So here is a picture of a salamander called axolotl. It has miraculous regeneration properties. If you cut off portions of its brain, it can still regenerate it. So now you may ask, how would I find out what aspects of the DNA are helping in regeneration so that potentially in the future we can incorporate those into other organisms? So if you want to do that, then if you can sequence the RNA of this organism when it is regenerating, then you'll get a quick signature as to which aspect of the DNA is involved in regenerative abilities. So this is actually a real application of RNA sequencing. So if you want to do other things, for example, understand gene regulation and so on, you want to know how much each gene is expressed. And the RNA is basically a measure for how much a gene is expressed. So among all the various star-seq assays, which basically convert different measurement problems into sequencing problems, the next most popular to the DNA sequencing is RNA sequencing. Okay, so the canonical nature of this problem and its important applications made us interested in this. So let me give you a quick primer of what is the relationship between the DNA, underlying DNA, and the RNA. So here is an example of a DNA string. So it's a continuous string, and these boxes are actually conceptual, so they don't actually exist. So it's a continuous string of alphabets over ACTG. Now you see that there are some boxes called exons, some are called introns. So this is a small snapshot of a genome, which is called a gene, which is involved in protein coding. In this, what happens is only these selected portions of exons, called exons, they come together in combinatorial ways to create an RNA. So here is an RNA transcript. So these RNAs are also called RNA transcripts because they are the output of transcription. So here is an RNA transcript, which is just a set of exons which have come together. So the RNA basically contains portions of exons pasted together in interesting ways. So here is one transcript from this gene. Here is another transcript from this gene. So as you can see, these two RNA transcripts are structurally correlated because they're coming from the same gene. So this means that there could be long repeats. For example, the third section is identical in the two. Not coincidentally, but structurally, because they're coming from the same RNA. So this is an important fact that different RNAs coming from the same gene share a structural correlation. So what is my role in this problem? So I come here because of the following reason, that you cannot sequence an RNA, so for example, each RNA is of length 10,000. The current sequencing technology does not allow sequencing the whole RNA in one shot. So what you do is you fragment this RNA into small pieces, and you can sequence each fragment. Now, once these fragments are sequenced, like here, for example, so you have a cell, and the list of transcripts in the cell is called a transcriptome. So in this cell, there are these two transcripts, each one repeated at different levels. For example, one may be related to a growth RNA, and which is expressed at some level. Another may be a skin cell RNA, for example. So it's expressed at a different level. So now what happens is, when you fragment this, you sample small pieces from this randomly. So for example, you may sample the piece TTC from the second RNA. And you get this in your digital version in your computer, which is called a read. So you sample this again and again, you get many different reads. So you had an original list of sequences, which is the list of transcripts or the list of RNA. And then here is your list of reads, which is the substrings from this original strings. OK, so all that is presented to the computer is just a list of reads. You do not know each read which RNA it came from. You also do not know which location inside the RNA it came from. So the set of reads is fed to the software called assembler, and the assembler basically takes these reads and tries to reconstruct the list of transcripts. So this is the portion that I'm interested in, how to develop the assembler. So this is a software, this is basically, the assembler is a software program which takes in these reads and creates the transcriptome. And there are many existing assemblers. Some are genome guided, which means they use the knowledge of the DNA to reconstruct the RNA. Some are de novo, which means they don't use the DNA to reconstruct the RNA. So they are basically side information free. So these are all the names of popular assemblers that are used. OK, so if these are the popular assemblers, then we may ask, why do we need to study this problem? Because the popular assemblers diverge significantly when you feed the same input. So here is a graph from this paper. So what we do is we take the set of reads. So to give you a size of scale, there are probably tens of millions of reads in the sequencing output. And these reads are fed into the software, and the software tries to reconstruct a list of RNAs. So if you feed the same input to these three popular software, then you see, so you ask, how much do they agree? So this is a Venn diagram for what sequence do they agree on and what sequences do they disagree on. So you see that the overlap between all of this is around 10,000, but the independent portion is really large. So the algorithms do not really agree on the list of reconstructed transcripts. So here we ask this fundamental question: is this because of an informational bottleneck, which means I do not have enough information to reconstruct the list of transcripts? Or is it a computational bottleneck? Because I have only so much time, I cannot really reconstruct it accurately. Or maybe it's neither. Maybe there is no information bottleneck and computational bottleneck, and a smarter algorithm can actually reconstruct them more accurately. So this is the question we asked, and here are our main results. We first quantified the informational bottleneck for RNA assembly using techniques from information theory, and then evaluate what is the quality and quantity of reads when reconstruction becomes feasible. So when do you have enough information to do reconstruction? So this is a kind of a traditional information theory question. It arises all the time in communication. You ask, when does a decoder have enough information to reconstruct the transmitted sequences? So we here ask a very similar question: when is there enough information to reconstruct the transcripts? So we quantify this informational bottleneck for RNA assembly. And then, so once we have characterized the informational limits, our next approach is to develop computationally efficient algorithms that approach this informational limit. So this is, again, drawing inspiration from communication theory, where the separation between the information theoretic limits and the computational expense was established by Shannon. So what we do is we go ahead next and develop a linear time algorithm that achieves near optimal performance for RNA reconstruction. Finally, we take these theoretical ideas and construct a software package that can deliver significant performance for transcript reconstruction over state-of-the-art algorithms. So the key ideas here all come from information theory, graph theory, network flows, sparse signal recovery, and finally software engineering to make this into a real software. So this project involved a significant overlap between ideas from communication and network theory and applications of them into this biology problem. I will illustrate the basic ideas as we go on. So here is a mathematical formulation of this problem. There is a set of transcripts, so they're sequences, and these transcripts have relative expressions. So some transcript may be expressed much more. Here, for example, transcript 2 is expressed at level 0.3, whereas transcript M is expressed at level 0.08. So in general, there is some relative expression levels of these transcripts. And they may share some common regions. For example, the blue region highlighted here is a common region between the first two transcripts. So we ask at what quantity, which means the number of reads, and at what quality. Each read, I said, is a substring of the original RNA. How long is this substring? At what point is reconstruction possible? So in order to solve this problem, we take the information theorist approach, which is to take a limit as n goes to infinity, which is when there are many, many reads, but the read length is fixed. So this limit makes sense because, as I showed earlier, the technological trend is that the sequencing cost is going down very fast, which means you can take a lot of reads very easily. And in fact, all present RNA sequencing experiments do have a lot of reads. In fact, hundreds of millions of reads. However, the read length, the substring length that you get from the sequencing technology is fixed and small. In the recent technology, it's 125. So the length of an RNA is 10,000 or 15,000, whereas the read length is 125. So we want to know, at read length 125, is it possible to reconstruct the RNA or not? Maybe there is not enough information when the read length is 125 to reliably reconstruct the list of RNA, in which case it will explain why the different algorithms are diverging. So this is the first theoretical question we ask. And the basic thing is we want to look for a phase transition on L, on the read length. So when read length is 1, right? So what does it mean? It means you get to know all possible length 1 substrings of the transcriptome. In this case, if the length 1 substrings are basically how many A's, how many T's, how many C's, how many G's are there. It sounds like it's almost zero information. There is no chance for you to reconstruct the transcriptome. But an interesting aside is in 1880, this was one of the first information that was got about the DNA, is that the number of A's is equal to the number of T's, and the number of C's is equal to the number of G's. And then this is how later Watson and Crick determined that the bases should pair together, because there must be some reason for this symmetry. So there is some information when L equals 1, but not enough to reconstruct a transcriptome, clearly. When L is really large, of course, any reasonable algorithm should reconstruct. In fact, there may be nothing to reconstruct if L is bigger than the largest RNA. OK, so we want to know what is the critical phenomenon, which means what is the critical read length below which reconstruction is not possible, and then suddenly above which reconstruction becomes possible. So typically in information theory, we like to have a statistical modeling of the source. However, like I said here, the RNA is, in fact, we did that first. We first started with putting a statistical structure on these sequences. Let us say each RNA is produced IID from some source and so on. However, these models don't fit the real data very well. So we went back to an arbitrary sequence assumption. And here, the L critical, the critical phenomenon, will depend on what the actual sequence is. So let me give you an example for this. So here are two pictures. These two pictures. So in some sense, RNA reconstruction or DNA assembly is like solving a jigsaw puzzle. So you have a big picture, and all you get is small snapshots of this big picture. So let's say you have this flower picture, and you got small snapshots or like small pieces of the jigsaw puzzle that you want to put together. What happens is when you have pieces from this puzzle, so among these two puzzles, if you ask which is the harder puzzle to reconstruct, the left one is harder to reconstruct. Because if you see a piece which has some flower petals and some blue, you cannot easily localize where it belongs to. Whereas if you see something, some piece over there, it's very easy to localize where it goes to. So in some sense, the complexity of transcriptome reconstruction, or in this case, a jigsaw puzzle assembly, depends on the repeat patterns inside the sequence. So this one, which has lower entropy, is harder to reconstruct. This one, which is higher entropy, is easier to reconstruct. This kind of goes in the opposite side of regular information theory, for example, in compression. OK, so the L critical that we want, for example, even this picture is easy to reconstruct if I have a really large rectangle. So if all the jigsaw puzzles are really big, then even this puzzle is easy to reconstruct. Of course, how hard a puzzle is to reconstruct depends on the size of the jigsaw puzzles. OK, so as I said, we want to now capture this L critical, and this L critical is going to depend on particular statistics of the sequences. In this case, the repeat statistics of the sequences. So here is the space of all read lengths. We want to establish this L critical. How do we do this? We have, just like in information theory, there are two things. One is we have to show an information theoretic limit, which means there is a regime where you're informationally limited. There is no way in this regime that any algorithm has enough information to reconstruct the original data. So, and then we have to propose an algorithm and then show that this algorithm can reconstruct as long as the read length becomes greater than a certain value. So we have these two bounds, and this sandwiches the value of L critical. And this is how we are going to, and these bounds are going to depend on the particular transcriptome. And we have a method which is also very efficient to calculate these values. OK, so so now I have to show you two things. One is how do we bound the performance of any algorithm, which means how do we establish informational bottlenecks. The second is how do we establish an algorithm that can reconstruct when you're beyond a certain critical read length. So here is an example. So here is a transcriptome with just two transcripts. The first transcript is S1, S3, S4. The second one is S2, S3, S4. The lines sitting on top of them are actually reads. Right? So let's say you have sampled reads. I've shown them uniformly here, but in general, they come from some random sampling. But let's assume uniform for now. And you see that these reads are sitting on top of these. So they are shown from which region this subsequence was obtained. Right? So for example, the red line sitting on top of S3 means that sequence basically is that substring of S3 coming from that region. As you can see here, the example I have illustrated is that the read length L is smaller than S3. So S3 is the common repeat across the two transcripts, right? So S3 is the common portion. It's the same in the two transcripts. The other things are all unique. And here, what we want to know is, if suppose you have this transcripts and you have this read length, is it possible to reconstruct this transcriptome by any algorithm, or is there an information bottleneck? We'll see that suppose you had this other transcriptome, which had these two transcripts, S1, S3, S5, and S2, S3, S4. So let me show this again, S1, S3, S4, S2, S3, S5, and I swap S4 and S5. This is a new set of two transcripts. These two set of new transcripts have the same set of reads as the original set of two transcripts. So if these two set of transcripts have the same set of reads, no algorithm can distinguish between the two, which means you're informationally limited as long as your read length is smaller than S3. Unless L is bigger than S3, these two transcriptomes can be confused. This establishes an information theoretic limit, right? So it says if your L is smaller than S3, then you do not have enough information to reconstruct the transcriptome. OK, so there is also other transcriptomes which you can confuse this to. For example, here is one with 4 transcripts. What are these 4? It's the same 2 and the other 2. So I show 2 transcriptomes. I'm placing them one below the other, S1, S3, S4, S2, S3, S5, and the swapped version. So now I distribute the reads between these 4 transcripts equally, right? So now all 4 transcripts have equal set of reads. This is also an equally well-defined explanation for the same data. So let us say there were 4 transcripts in the original transcriptome. Then these reads are fully explained by these 4 transcripts, right? So in general, the problem is not well defined. So given a set of reads, what transcripts does it come from? What sequences could have generated this is not a well-defined problem. However, sparsity, which means I want to give this, of course, there is always a more complex, for any explanation, there is a more complex explanation. So in this case, for a 2 transcript transcriptome, there is a 4 transcript transcriptome which can still explain it. By using Occam's razor, we say that the sparsest or the smallest set of transcripts that explain the data should be the right answer. So this is, again, a principle of sparsity, which has been widely studied in signal processing and so on. So here, the sparsity can say that this 4 transcript transcriptome is not a good explanation. However, the first 2 both had 2 transcripts each. It's not possible to disambiguate between the 2. So there is an information bottleneck. How do we get rid of this? In wireless communication, when we faced a similar bottleneck, when there is a fading channel, for example, you want to communicate over this fading channel, and the channel is varying over time, initially it was thought of as a bottleneck in the sense that the channel is varying, it's not a good thing. However, later it was shown that this diversity of channel variation can be used opportunistically. So we adopt the same philosophy here, and we use something called abundance diversity. So what is it? So let's look at these 2 transcripts. In reality, so the toy example I had created here had these 2 transcripts equally expressed. There were the same number of reads sitting on the first transcript and the second transcript. However, in reality, transcripts do not all come equally made. Different RNAs are expressed by the cell at different levels. It has been shown using data that the expression levels follow a continuous distribution. So in general, the expression level of 2 transcripts will be very different. So here I have shown transcript 1, which is lowly expressed, transcript 2, which is highly expressed, which means there are more reads coming from transcript 2 than reads coming from transcript 1. Suppose you have this. So this is what we call as abundance diversity. Different transcripts are expressed at a diverse levels. So here, if you say that this is an alternate hypothesis trying to explain this data, then this doesn't hold water because there is uniform coverage on this first explanation, whereas highly non-uniform coverage on the second explanation. So the second explanation is not as good an explanation as the first one, and therefore we can narrow down to the right set of transcripts, even if L is smaller than S3. So even if the reads are smaller than the S3, we are able to narrow down to the right set of transcripts because we are using the diversity of abundances. So in RNA sequencing, this abundance diversity was considered to be some kind of a curse because the transcripts that are lowly expressed need more reads before which you can reconstruct them. However, the quantity of reads, like I explained, is not anymore a bottleneck. You have many, many reads. You have millions, hundreds of millions of reads. The bottleneck here is the read length. And here, what is shown is if we can estimate the coverage of transcripts, then really the diversity of abundance is a blessing, not a curse. OK, so I gave you the basic idea of how to obtain certain types of informational limits. I have not gone into the details, of course. Now I'll give you some hints of what kind of algorithms are used to reconstruct the transcripts from the reads. So recap, you have a set of reads, and the algorithm needs to take in these reads and output a list of transcripts. So what do we do? How do we do this? So you take the reads, and then once you have these reads, so here there are some six reads. So what you do is, how do we make sense of, how do we reconstruct a DNA or RNA from this list of reads? So the basic idea of what you do is to look for overlaps. So here you see that these two, the reads on the right, are basically, there is an overlap of ATTC between them. So then you expect that they probably came from the same region of the RNA. So you say, yeah, maybe they're adjacent. So whenever two reads have a certain overlap, then you say that they probably come from the same portion of the RNA. So we do this again and again. So here there is another two reads, and there is an overlap TCCA between them. So you say, yes, maybe they come from the same portion of the RNA. And then you can keep doing this till you get a graph. So you take the set of reads, and you converted it into a graph. So we call this a transcript graph, because potentially transcripts come from this graph. So we do have some more information than just this graph. We know, for example, how many times each subsequence appeared. For example, you may see that the subsequence TCCA appeared in the reads at frequency 0.3, whereas that substring ATTC appeared at frequency 0.1. So what you do is you take these reads, you measure how many times each one sits on this graph, and then you get some numbers on this graph. So you not only have a graph now, you have a weighted graph. So and the weights are based on relative expression levels of these various reads. OK, so like I said, this graph has many overlaps. So you simplify the overlaps and just look at the unique portion. So here the unique portion is marked in red, and then you create a smaller graph. So we call this condensation. We can condense this graph to just get the unique portion, and we get a graph like this. So this is the small graph that we can obtain from the set of reads that we started with. Now, we need to reconstruct transcripts. We don't need to reconstruct a graph, so we have to see how to reconstruct a transcript. So if this is the graph that we had before, and what you see is different paths in this graph can correspond to transcripts. So for example, the path GATTCG could be one sequence, and another sequence could be ATCC-ATTCG. So this is one way to reconstruct a list of paths from the graph as transcripts. So here is one naive algorithm. The algorithm will list out all possible paths in this graph and say these are the list of transcripts, potential list of transcripts. So the thing about this graph is the real transcripts are paths in this graph, but not all paths are transcripts. So we need to figure out which are the real transcripts and which are not the real transcripts. So in fact, this algorithm I call as a naive algorithm, but this is one of the popular software. In fact, in the intersection figure that I drew, there is an algorithm called Scripture, the software called Scripture, which had the largest bubble. So it basically outputs all the paths. So OK, so what do we do? Right? So earlier, if you remember, when we got confused between two possible transcripts, we said that what gives you the clue to reconstruct the right set of transcripts is the abundance diversity. The relative frequency of expression gives you a clue. So let's consider this graph, S1, S3, S4, S2, S3, S5. This comes from that transcriptome. So now what happens is, so there are two possible ways to go through this graph. One is to say S1, S3, S4 is a path, S2, S3, S5 is a path, right? Another way to say is S1, S3, S5 is a path, S1, S3, S5 is a path, and S2, S3, S4 is a path. So which of these two ways is correct, right? So that's the problem, and what we do for this is to use the relative frequencies. So for example, if you have the relative frequencies of these various nodes as 0.1, 2, 1, and so on, now you see that the path which goes through this has basically a continuous abundance distribution, which is 0.12 going through all of these nodes, and 0.88 going through the other nodes. Whereas the alternate explanation goes through nodes of very different abundance, and therefore not a valid explanation for the graph. Okay, so here is the first idea, is to use the continuity of copy counts, or continuity of abundances. So 0.12 has to be paired with 0.12, 0.88 is to be paired with 0.88. So here is the first idea, and turns out this idea some people have already used in the software. So what happens is you get more complex splice graphs. So for example, so these transcript graphs are also called splice graphs because they have a certain biological meaning. Okay, so here is a graph where there are 3 nodes incoming and 3 nodes outgoing. And I've put some numbers on them. And if we try to use the same heuristic that I tried before, I'll look at 12. So there is 12, 5, 7, 15. So none of the numbers match exactly. So I may say 12 is closest to 15, so I'll pair those two, and that's a path. I may say 9 is closer to 7, and those two may be a path, and 6 is closer to 5, and those two may be a path. So this is what existing algorithms will do. However, there is a better explanation. The 12 is just 5 plus 7, and the 9 plus 6 is 15, right? So if I output these 4 set of paths, they explain the data perfectly, right? So this is the smallest set of paths that explains this data perfectly. So my claim here is now there are 4 transcripts, S1, S3, S4, S0, S3, S5, S1, S3, S6, S2, S3, S6, and each one having the corresponding abundance levels such that they add up to create this graph. So in general, we are given values on nodes and edges, and we need to find that flow which goes on the fewest paths. So here is again main relation to work in networks and in information theory is network flows. So how network flows tie together to this problem and how that can be used to solve this problem. So in general, as I said, the principle is to find the smallest set of paths that satisfy all the node and edge abundance values. So this problem turns out to be of importance even in network routing, snooping, and in societal networks. So let me tell you what the problem is. The problem is called how to split a flow. So what is a flow? Suppose you have a network. So nominally, we think of a flow as just like if water is flowing on this network, then there is a natural conservation. Whatever comes in has to go out. So there are 2 ways to represent a flow. One is to represent the value of the flow on each edge. What is the value of flow on this edge, and so on? So here, for example, there is a conservation constraint that 0.12 plus 0.88 is equal to 0.12 plus 0.88. Right? So this is called an edge flow. You specify the value of flow on each edge, and these values satisfy the conservation. Whatever comes in goes out. Total in equals total out. So there is, however, another way of defining a flow, which is to specify how much flow goes on each path. So here are 2 paths, and on the first path, flow of value 0.12 is flowing, and on the second path, flow of value 0.88 is going. So this is again, this may look like an equivalent way. However, it turns out, given a path flow, it's easy to find an edge flow. It's unique. However, given an edge flow, there is no unique path flow. There could be many path flows that satisfy the same edge flow. This is just like the example that I illustrated earlier, where 2 transcripts can be explained also by 4 transcripts. So these 2 paths could also be explained by 4 paths going all over the place. So given an edge flow, what we want to do is we want to find that path flow with the fewest number of paths. Okay, so why is it relevant to networks? Because in network routing, for example, let's say you have some network flow. You want to send some packets from one source to the destination. You want to split it into as few packets as possible and send it on as few paths as possible. So this problem turns out to be useful in network routing. It's also useful, for example, in societal networks. For example, let us say you're studying a transportation network, and all you observe is traffic on the edges, which means I observe so much traffic on I-5, on I-20, and so on, but I want to estimate how much traffic is going between San Diego and San Francisco, or how much traffic is going between Seattle and San Diego. So you want to infer end-to-end flows by looking at local values. So this is the same problem as given an edge flow. So the edges are the roads here in this transportation network. Given an edge flow, how to get the path flow. OK, so that's the relation to network flows and network information theory. And here is so turns out these people have studied this problem, the network theorists, and they've shown that this problem is NP-hard. OK, so the problem is NP-hard means that there are hard instances of the problem to solve. But we have to find out whether our instances that we are facing in the biology are these hard instances or are they the easy instances. So we go and take a look at a closer look at these hard instances from the hardness reduction. And then what happens is in these hard instances, basically almost all the paths have the same flow. So path 1, path 2, path 3, path 4, they may all have flow value 1. Path 5, path 6, path 7, they have flow value 2. So in these hard instances of flow decomposition, it turns out that most paths have the same flow, or they have only one of two values of flow. This is equivalent to saying that in our case, most transcripts have the same expression level, which is totally not true. Different transcripts have different expression levels. In fact, two different transcripts will have almost surely a different expression level. This is not characteristic of the biological problem. So we went and dug this deeper, and our basic result is that if these abundances are generic, which means the relative expressions are drawn from some continuous distribution, then no two will be identical. And we propose a provably correct algorithm that reconstructs the list of paths under a certain condition. And the algorithm does this reconstruction in linear time, not just in polynomial time under this condition. And it turns out really nicely that this condition is satisfied in the biological data. So the algorithm is an iterative algorithm, so you want to resolve the set of paths globally. But looking at a local snapshot of a node, it tries to resolve what paths could go through this node. And it uses the fact that different paths in general should have different abundances, which means we use abundance diversity. So when designing this algorithm, we have to deal with these issues of error propagation. For example, if you make an error when you decode a node, that may propagate to all other nodes. You, for example, wrongly decode a path, you may decode all other paths wrongly. So we have a fix for this. What we do is we decompose a node only when we are sure. So this is very similar to erasure decoding of codes. So only when you're sure, we decompose a node. If we are unsure, the node is left as it is, and other nodes are tried before coming back. So the algorithm solves the network like solving a Sudoku puzzle. You put in a number where you know you're sure, and then that helps you solve other numbers. So let me give you an example here. So here is a network, and there is some numbers on this. Is a weighted graph. And let's say you try to decode node 3. So node 3 has some paths going through this. Turns out the algorithm can decide that it's hard to decode this node because none of the numbers match up. A doesn't equal C, B plus C doesn't equal A plus B. It's very hard to decode this node. So now the algorithm goes and looks at another node. Let's say it goes and looks at node 4. And then node 4 is a somewhat easier node because A plus B is coming in and A and B are going out. So the only way this can happen is 3 goes to 6 through 4 at frequency A, and 3 goes to 7 at frequency B. So we can resolve that portion of the graph, and then we can come back and now look at node 3. Now there is an A matching up with an A. And this can be, so once you solve that, then the B plus C has to split into B and C. So you can use this basically to reconstruct a list of paths. And basically, if you track the graph, you will see that the A is going 1, 3, 4, 6. And the algorithm reconstructs 1, 3, 4, 6 as a transcript, and similarly the other ones. So the algorithm can reconstruct the list of transcripts using these numbers as keys. And these numbers, locally, the fact that no two of them are the same helps you locally resolve certain things. And there is a condition under which this iterative algorithm is correct. And we can characterize that. So like I said, so I presented a way of finding bounds on algorithms. I also presented a way, an algorithm, which reconstructs in linear time. And these two bounds, if you evaluate these bounds on real data, for example, on a mouse transcriptome, they match exactly, which means the algorithm we have is optimal for this data set. So here, surprisingly, informational sufficiency, which means whenever you have enough information to reconstruct, you also can do it really fast. Informational sufficiency implies computational efficiency. So the original problem was NP-hard under general instances. However, the instances that are easy to solve, where you have enough information to solve, are also easy to solve. So it's a very nice alignment of information theory and fast computation. OK, so what is the value of L critical? So this is the theorist's dream. So we have exact theoretical characterization establishing the critical phenomenon. However, the value of L critical turns out to be 4,000, which is a practitioner's nightmare, because the L critical is 4,000, whereas the real values of L are 125. So what do we do now? What can we do at practical values of L? So initially, we thought maybe this explains the discrepancy between the different algorithms, why algorithms are not able to agree on the output. However, we asked a different question, which is you fix a read length and ask so you have a set of transcripts, right? Let's say you have 10,000 transcripts. You ask what fraction of these transcripts can be reconstructed at a given L. So this is partial reconstruction, right? So in information theory, we take these limits and we say with epsilon error and epsilon goes to 0 and so on. So here we take a similar approach and we say that we only want a fraction of transcripts to be reconstructed. What is the maximal set of transcripts that can be reconstructed correctly? So here is the information theoretic bound. So it's an upper bound which says that no algorithm can reconstruct more than this fraction of transcripts as a function of read length. For example, when read length is 100, it says that no algorithm can reconstruct more than 94% of the transcripts correctly. That doesn't sound very bad. So what does our algorithm do? So our algorithm can reconstruct, so it says at read length 100 again, our algorithm can reconstruct nearly 94% of the transcripts correctly, very close to the upper bound. So initially, we were thinking maybe it's a theoretical artifact, but we can show that any algorithm that does not use abundance diversity has to have a performance lower than this curve, which means, for example, at read length 100, no algorithm can reconstruct more than 55% of transcripts correctly if they do not use abundance diversity. So this is a very nice result showing two things. One is our algorithm is near optimal. Second is any algorithm that does not use this information properly will be highly suboptimal. So it shows separation between existing algorithms and our algorithm. Again, like I said, varying abundance is a blessing, not a curse. So we evaluated, so we built a software. We got very excited, and we built a software package out of this. And in order to verify whether the software is doing correctly, we need to know the ground truth. What is the true list of RNAs? So we use detailed simulations of reads from the human chromosome 15, RNA reads from the human chromosome 15, and we have a certain number of transcripts, and they're sampled using some log uniform distribution. We're modeling realistic error rates, realistic read lengths, and scaling the reads appropriately for this problem size. So here is our practical performance. We ask, given a true list of transcripts, we sample reads from this, and then we reconstruct using our algorithm, and also using a competing algorithm called Trinity, developed at the Broad Institute at MIT. OK, so what happens here? So here, I've fragmented this picture by the coverage depth of transcripts. So for example, when the transcript coverage is 100, which means that on this transcript, on each position in this transcript, there are 100 reads sitting on an average. So these are well covered. So like I said, the transcripts, each transcript occurs at different ratios. So some transcripts will have more reads sitting on them. Other transcripts will have less reads sitting on them. This is a finite number of reads. So some of the transcripts will have very little reads sitting on them. So we look at transcripts which have more than 100 reads sitting on them. We see that our algorithm, we call it Shannon RNA, because it's an information theoretic RNA assembler. And we see that our algorithm reconstructs nearly 98% of the reads, of the transcripts, whereas Trinity reconstructs less than 80% of the transcripts. So even though our algorithm was derived at the limit of n tends to infinity, which means at the high coverage regime, we have many, many ideas to make this algorithm work at the finite coverage regime. For example, here, you see that Trinity reconstructs 55% of the transcripts, whereas we are able to reconstruct nearly 83% of the transcripts, even when the coverage depth is low. So the ideas involved to make this work are actually, again, ideas from network flow, min-cost network flows, maximum likelihood smoothing, randomized algorithms for sparse recovery, and so on, are used to make this algorithm adaptable at finite n. So, and also, the other thing is if you output all possible paths as transcripts, then you will do very well on this metric, but very badly on the other metric, which is the false positive. We should not output a transcript which is not a true transcript. So what fraction of transcripts are not true transcripts? We do better than Trinity because we output fewer transcripts which are false positives. OK, so here is a summary of the RNA assembly problem. We quantified the information theoretic limits for RNA assembly. And information limits suggest that there is a huge gap between existing algorithms and the theoretical limits. And we close this gap by proposing a new linear time algorithm. And there is algorithmic innovation here using ideas from network flows, maximum likelihood estimation, and sparse signal recovery. And finally, we implemented software with significant performance advantage over existing state-of-the-art software. So currently, we are working on optimizing this for a multiprocessor implementation so that the scales of data that we can deal with. So currently, the algorithm can work with 10 million reads, but it needs to work with 100 million reads to really beat existing software on computation time. So the algorithm is nicely parallelizable. So we are just, it's a matter of writing the program. Currently, we're doing that. OK, so I've also done some work on wireless networks. And the connection between this problem and the networks problem comes from this underlying use of network flows and information flows in networks, which I studied in my PhD thesis. So in the wireless network, so just like I showed the sequencing figure, the motivation for studying the wireless problem is that the mobile data is going up tremendously. And at this scale, interference becomes a key bottleneck. And we want to understand when does interference limit your capacity of a network? So the capacity of wireless networks is a long-standing open question. And even when sometimes the theoretical understanding exists, engineering implications are unclear. And in our work, we basically studied this problem, a general multi-source, multi-sync network, which means you have a network with several sources, several destinations. Each source wants to communicate to its destination. And for this problem, we proposed a new scheme involving interference alignment and network coding, followed by routing. And we showed that this scheme achieves the approximate capacity of this network. So this basically solves a long-standing open question in wireless network theory, which is how to optimally communicate information when there are multiple source destination pairs in a wireless network. We also have engineering guidelines, which means we show how to translate these ideas into potentially engineering ideas. Like, for example, the proliferation of wireless networks is mainly due to layering, which means different layers perform different tasks. And these layers can be designed independently. So in our work, we show how this scheme, the near optimal information theoretic scheme, has a natural layering strategy. And so let me quickly go over what I mean. So in the current, so for example, let's say this is a current wireless network. So in this network, the basic idea is to schedule orthogonal links. The current networks mostly do this, schedule orthogonal links and then do routing. Like, for example, you schedule this link and this link because they don't interfere with each other. And then you similarly schedule many of these links. And then finally, you route information through the network. So basically, the idea is to schedule orthogonal rings and then route information. So this kind of strategy, we can show, is provably far away from the network capacity. So these schemes don't do very well in terms of network capacity when you have many source-destination pairs or many nodes in the network. So what we do is to use this technology called interference alignment. So interference alignment is a method for managing interference. However, it was developed in the context of simple networks, single hop, set of sources directly talking to destinations. A very nice technique, but only in the case of single hop networks. And the nice thing that they showed is interference need not be a big bottleneck, because if you use this strategy, which is very nicely developed, very nicely coordinated across the different sources, then basically you can get interference, almost half the interference-free rate, even when there is interference. So what we do is we take a general network, we find a subset of links that can be scheduled using interference alignment. Like, for example, this link, this link, this link, and this link. So 4 links can be scheduled simultaneously in this example using interference alignment. And then we schedule different links using maximal matchings in this graph, which are basically related to alignment. And then we route information through this. So the natural question is this joint interference alignment and scheduling and routing, how good is this scheme? And our main result says that the rates achieved by this scheme are within a log k factor rather than a factor k from the information theoretic bounds. So when you're provisioning a network with k users, you have to scale the network resources as order k. So when you're scaling the network resources as order k, taking a hit of a factor log k is a very small factor. So this is our main result in network information theory. And this comes by proving some results for what we call as polymatroidal networks. So polymatroidal networks are somewhat like wireline networks. So they have capacitated edges. But the capacities interact with each other just like in a wireless network. For example, in a wireless network, if I'm talking to user A, my communication rate to user B is restricted. So there is an interaction between the edges that happens in a wireless network. And we can capture that using some submodular set functions and what we call as polymatroidal networks. And what we show is on these submodular networks or polymatroidal networks, routing is near optimal. And we also show that given a wireless network, we can find an equivalent polymatroidal network, which is equivalent in terms of the information theoretic capacities. And if you can achieve any rate on the polymatroidal network, you can achieve a similar rate on the original wireless network. So this has the independent mathematical interest as it generalizes many results in graph theory, like the Max-Flow-Minkert theorem, polymatroid intersection theorem, and so on. And finally, in order to make these techniques practical, so we developed more practical interference alignment techniques. So interference alignment is a very key theoretical technique now used in information theory. However, there are many barriers to practice. For example, large channel diversity is needed. So the channel needs to change quite often. And also, at finite SNR, the performance is not very good. So we propose something called interactive interference alignment, which is when you can use the fact that a wireless channel has natural interaction. If I can talk to you, you can talk back to me on a general wireless network. However, in the wireless models, typically graphs are assumed to be directed and acyclic because information flow is easier to study there. However, when we model this natural interaction of this channel, we can show very simple alignment schemes, linear, small block length, very good finite SNR performance, which do very well in practice. So finally, I've also worked a bit on streaming codes for distributed computation, where normally error correction codes are designed to operate on blocks of data. So you have a block of data coming in, and then you process it, and then you spit out a block of data. However, what if bits arrive one at a time? Like, for example, when 2 cores inside a deep submicron network, 2 cores, for example, are communicating with each other, then the bits that I'm going to spit out next, I do not know a priori. I'm waiting for the computation of the other core before I can spit this one out. So this happens in distributed computing, and we build new error correction codes, which are causal codes, and which have very good distance properties. So earlier it was known that these codes exist. However, no efficient construction was known. In work with at Microsoft Research, I basically showed the first efficient construction of tree codes. So finally, I'll wrap up with some concluding thoughts on future work. So one of my main focus is to understand the foundations of computational biology using the information theoretic lens and the network theoretic lens, and developing algorithms that can basically achieve these information and network theoretic limits. And the other areas that I'm also actively interested in are interference management in wireless networks, network capacity, and practical algorithms to achieve them. And finally, in information theoretic privacy, primarily in applications to computational biology. For example, when you're distributing data sets, you want to make sure that the data set has a certain utility, but it also protects the privacy of the people whose data is being put out. So this is, again, an interesting area of research. Finally, I would like to thank all my collaborators, and thank you all for listening.
Maybe one question? Sumit? Josh?
Proceed.
So from the sequences from the information side, how does the initial work relate to typical sequences and high probability sequences? Because you don't seem to be taking that discussion into
Yes, so it's a very good question. So the question was, how does the sequencing information theory relate to typical sequences and so on? So like I said, initially when we started working on the RNA sequencing problem, we developed the RNA sequencing theory for a stochastic model. So when you assume stochastic models and you take certain limits, then you can start talking about typical sequences. Which of these happen typically with respect to this source distribution, and so on. So however, like I said, the models, the stochastic models for the RNA sequencing problem are not well developed. The simple IID models, or even Markov chain models for RNA sequencing, show that very simple algorithms are optimal, and in reality, they are far from optimal, because the sequences do not display simple Markov properties. Like I said, for example, if one block is exactly the same as another block in a sequence, then none of the Markov chains capture this thing. So that is why we had to go away from stochastic information theory to worst case or combinatorial information theory. So that's kind of the relation to typical sequences. However, I should say that the information theorist thinking is in terms of typically what happens. And this was basically reflected in our work, because the NP hardness is a worst case result, which says that, if you take the worst case list of transcripts, then there is no way to assemble them in linear time or in polynomial time. However, we see what we said is, what is the typical occurrence in a real data? So this is empirical typicality. So empirically, what is the data distribution? And we use that typicality in order to reconstruct the list of transcripts in linear time.
Yeah, the continual distribution exploitation of that.
Yes.
And also throwing away the max probability sequence
Yes.
reminds me of the fact that you
Exactly. So that is the context where we use the typicality. Any other questions?
Do you know what the value of your critical value L is for any other animals besides the mouse?
Yes, so I've done it also on the humans. And at read length 100, again, you can reconstruct nearly 92% of the transcripts. So very similar. So the mouse and human are very, very similar. We have not done it on other so we are working on yeast currently.
So generally, you think it'll be around 4,000 for most of them?
So like humans, mammals will be around 4,000. And however, plants, for example, have very large repeats. So they may be even more. And bacteria are very information efficient. So they do not create repeats. So they will be even smaller. So that's kind of the, so it's surprising that the hierarchy of, for example, the DNA size doesn't directly relate to the evolutionary complexity. Like plants have much larger DNA than humans. So, yeah.
All right, thank you very much.